Okay, first up, a couple of reminders. Overdue assignment deadline, 3:20 today. Make sure if you want it marked that it's in by then. Second reminder, okay, you're writing your final exam on Thursday at 9 a.m. So that's January 25th, and you guys are writing down the math wing. Hang on, I gotta get that room number again, room name again. January 25th, Thursday. And you guys are writing in St. John. I still don't know whose room that is, but it is down the math wing. So go.
I'm sure that's Miss Olszewski.
Miss Olszewski's room? Okay. So, probably Miss Olszewski's room. Sounds like it. Okay. If not, go down the math wing. Look for St. John over the door. Okay. I mean the name, not the actual guy. Okay. That'd be cool, but also scary maybe. I don't know. Just make sure you're looking for that. Okay. Do not go to the theater where 5 sections of regular Science 10 are being written because you will not find your test there. Okay. The AP is just writing down the math wing. So, what do you need to bring with you to the test?
2 pencils.
2 pencils, 2 pens.
Calculator.
Calculator has what in it?
And my smile.
Mario.
Mario?
Nothing.
Fresh batteries, or it is fully charged, whichever applies to your calculator, okay? So that if I need to clear it, it is the type that requires clearing, okay? That I can do so and not have to take it, put it in my pocket and walk around everywhere, okay? Until I get it reset. So make sure that you have all of that stuff with you, and then of course, you want to, something to drink, okay? Or an exam appropriate snack. Okay, that's fine. Just make sure, okay, that you have all of that stuff with you when you go to the test. What can you not bring to the test?
Fried chicken.
Yeah, well, I mean, apparently you can bring fried chicken, but please don't.
Or some KFC.
Yeah, exactly, yeah.
Oh, that's unsettled.
Yeah, I know.
KFC. I can't do pop.
I can't eat Kentucky Fried Children. I mean chicken.
I can't do.
Okay, so make sure, guys, that you do not bring any electronics other than your calculator. Okay, your phone needs to be in your locker, your car, left at home. Okay, your smartwatch. Okay, especially if you have one of the 4G, ones that's cellular enabled. Okay, your earbud, wireless earbuds. Okay, anything like that that can transmit or receive within the room needs to be left outside of the room. Okay. All right. So let's start off with questions from you. What are some things that we want to go over today, Alex?
Acceleration question.
An acceleration question, okay.
This guy.
How am I supposed to roll this? It's like old stuff, like Quinn. Yeah.
How did they figure that out? How did they learn how to cheat it?
It's good as second anyway, so it doesn't matter.
Okay, you should be writing this down, guys. I shouldn't hear talking.
I'll take both. I am ready.
Okay, so in this one, looking for the initial velocity, okay, of an object that accelerates at negative 4.5 meters per second squared for 12 seconds and reaches a final speed of positive 6.1 meters per second. Probably on an exam, I wouldn't put the negatives and positives in there. I might say it slows at a rate of 4.5 meters per second squared, okay? You would have to know that that would mean negative, right? All right, give you a couple minutes on that one. All right, let's have a look at this one. So here's what we know. We are looking for the initial velocity. Okay, so VI is our question mark. We know the acceleration to be negative 4.5 meters per second squared. We know the time to be 12 seconds. And we know the final velocity to be positive 6.1 meters per second. Okay, A equals VF minus VI over T. And we are trying to solve for VI. This is the trickiest manipulation of this formula. Okay, so first thing I want to do is move T because it's not attached to VI. Okay, so I'm going to multiply both sides by T. Whoop, that's not what I wanted. I'm going to multiply both sides by T. Then what I'm going to do, because I don't want to solve for negative VI, is I am going to add VI to both sides. Okay, that's going to move it away from there. And then I am going to subtract T times A over to this side. That'll leave me with VI. Okay, so now I can plug in my numbers and solve. So I will have 6.1 minus 12 times negative 4.5, okay, which should give me a nice large positive number.
Look at all these formulas on the test, right?
Absolutely, they're on the back of your periodic table.
That's true.
I got it.
Okay, so 6.1, okay, minus 12 times negative 4.5, right? So my initial velocity is positive 60 meters per second. Okay, since lots of people are asking, do I get all of this stuff? Okay, I'll show you what your Okay, so the back of your periodic table looks like this. You got your solubility chart. Okay, you have these common colors of aqueous ions, which you're not going to use, so you can ignore that part. Okay, you have your graphing equation. You have the mole equation. You have displacement's final position minus initial position, average speed, average velocity. You even have this final position minus initial position over time put in there. Average acceleration formula in there. Okay, force, which is not something you're going to use, but we talked about it, so it's there. The work formula, okay, how to calculate weight, which you won't use. Okay, your kinetic energy formula, potential energy formula, mechanical energy formula, efficiency formula, thermal energy, MC delta T, MLF, and MLV. Your constants, 9.81. Your heat capacities, water, ice, steam. Okay, I even give you the one for gold because I think you need it on there. Okay, you got your latent heats of fusion and latent heats of vaporization. Okay, and you've got Avogadro's number. Okay, everything that you will need for the test. If there's anything else you need, it would be in the question. Okay, so all of that stuff is on there. And then, of course, on the other side of that is your... No, apparently it doesn't want to respond to me at all. So, okay, you can play that game.
Mm-hmm.
Oh, maybe they can't. Yes, they can. I win. Okay. All right, what else? What next?
Can we go over the staircase thing from global systems?
Yep, we absolutely can go over the staircase thingy.
Also, the mole equation after that.
Okay, well, just hang on, we'll do one at a time. Okay, all right, so the staircase thingy could have a question like this.
Not a receiver.
Okay, so a staircase or heating curve is what we call it. Question, okay, would look something like this. How much energy is required to heat water at 13 degrees Celsius to steam at 125 degrees Celsius? Okay, so if we get a question like that, we know that we're dealing with the heating curve of water. So the first thing we do is draw the staircase thingy, which is called the heating curve of water. Okay, odd because it's not a curve, but that's what it's called. Okay, so we got a diagonal line, a flat line, a diagonal line, a flat line, and a diagonal line. Okay, that is the heating curve of water. The two flat lines occur at the melting and boiling points of water, 0 and 100 degrees. Okay, so what I have to do is mark on this staircase heating curve thing, okay, where I start and where I finish so I can identify how many and what steps I need to do in order to calculate the energy required. Okay, so I'm starting at 13 degrees. Since this flat line is 0, I'm going to mark right there as my starting point, slightly above it. Okay, 13 degrees. I'm going for 125, so that's going to be up here, slightly above the other flat part of the graph, which is at 100 degrees. Okay, so now I've marked my starting and ending points. Anywhere the line changes direction is the end of one step and the start of the next. Okay, so step 1 goes from here to there. Step 2 is here, and step 3 is here. Okay? So I've identified, I have 3 steps. Now I have to figure out what are those steps. Anywhere the line goes up, what's changing? The temperature. Okay, so I know that step 1 involves a change in temperature, which means a change in kinetic thermal energy, which means I use this formula that involves change in temperature. We do need a mass. You are absolutely right. 200 grams. Good eye. Okay. Coderre needs to drink more coffee very soon. Okay, so that would be step 1. In step 2, the line is flat. And step 2 occurs at 100 degrees Celsius, where water is doing what?
Changing state.
Changing state from liquid to gas. Okay? So when that happens, obviously, I cannot use MC delta T. There is no delta T. If I use that formula, it'll tell me no energy is required, and that's obviously not true. So I have to use the formula that is involved in change of state from liquid to gas. Okay, and that is latent heat of vaporization. Okay, and then in step 3, okay, I start with steam at 100 degrees, and I'm going for steam at 125 degrees. Line is diagonal, so again I'm using E equals MC delta T. Right? So now that I've got the steps figured out, now I can just do them. Okay? Just plug in the numbers. I don't have to manipulate anything. I just need to put the right numbers in. Okay? So as Rachel pointed out, we needed a mass, and we know what it is now. It's 200 grams. Okay? And that's multiplied by the specific heat capacity. What specific heat capacity do I use there? The one for liquid water. Right, which is 4.2. Okay? And how much am I changing the temperature? 87 degrees. I'm going from 13 to 100. Okay, so 87 degrees. That is where the most common mistake is usually made. Okay, so we've got 200 times 4.2 times 87 degrees. So we're looking at 73,080 joules. All right, so that's step 1. Step 2. Got to vaporize the water. 200 grams times the latent heat of vaporization, which is 2,260 joules per gram. So that's going to be a big number.
Is that going to be on the sheet that we...
I showed you. It is on there. Yeah. It's near the bottom, but it's there. Okay, so that's going to be 452,000 joules. Okay, and then in step 3, I've got to heat it up again, so I've got to change the temperature, 200 grams times, now what heat capacity do I use? One for steam, 2.02. joules per gram degree Celsius. And the temperature change for this one is from 100 to 125, so 25 degrees Celsius. Okay, so plug that one in. All right, so we're looking at 10,100 joules. Okay, what do I do with those three numbers?
What was the number you used in the second part of step 2? Like not the mass, but
That's the latent heat of vaporization for water,
is that
LV.
back your periodic table?
It is, yeah.
And then what's 333? What is that?
That's the latent heat of fusion.
Okay.
That's the one for changing it from solid to liquid.
Okay.
Okay, so we got those three numbers. What do we do with them? Add them up. Okay, so I take that one plus that one plus that one, and I get 535,180 joules. Okay, so I would probably on a test give you a mark for that, 2 marks for that, 2 marks for that, 2 marks for that, 1 mark for that, for a total of 8 marks. Okay. Okay. All right, someone said mole equation, yes?
Yes.
Okay, what kind of mole equation?
I'm only have 5 more mass as well. I don't know.
Avogadro's ones.
One of the Avogadro ones? Okay, that's kind of what I thought.
Yes.
Yeah, dog.
I will not get my shot in.
Darn, I'm sorry.
Okay, how many molecules of iron 3 hydroxide are in 350 grams? Okay, so first off, okay, we're looking for the number of molecules. Okay, that is our question mark, okay, for this one. Okay, they do tell us the mass, it's 350 grams. Okay, and they do tell us what the material is. It is iron 3 hydroxide. Okay, so I'm going to have to bracket the hydroxide and put a 3 outside of that. That will be my formula. Okay, so I'll have 55.85, 1 iron, okay, plus how many oxygens? 3. Okay, so 16 times 3, plus how many hydrogens? 3, so 1.01 times 3, so 3.03. Okay, so we punch that in, we got 55.85, okay, plus 16 times 3, which is 48, okay, plus 3.03. Okay, so we've got 106.88 for our molar mass, grams per mole. Okay. What can I find right now since I have both M's? I can find the number of moles. So that's what I should do. N equals little m over big M. So I'm going to take that 350 grams, I'm going to divide it by 106.88, okay, grams per mole, and I'm going to be left with moles. Okay, so 350 divided by 106.88, whoop, 0.88. All right, we're looking at 3.27 moles. I'm going to keep all those decimals in my calculator. I'm just not going to write them all. Okay, here's where we use Avogadro's number. Okay, Avogadro's number tells me the number of molecules in how many moles? 1. I have 3.27. What do I do with Avogadro's number? Multiply it by 3.27. Okay, so the number of molecules is going to be 3.27 times 6.02 times 10 to the 23.
And don't you have to keep all those numbers in your calculator?
I am keeping all the numbers. I'm just going like this. Answer, okay? And then bracket 6.02 E23.
Do you have to bracket it?
You do have to bracket.
1.9.
Okay, 1.97 times 10 to the 24 molecules.
I didn't like write that down. I'm never going to.
Okay. What else?
Wait, Jackson.
So on the
Can we do one
final
more of these?
exam,
Guys, hush please.
I was confused on number 15 on the final exam.
Read it to me, I don't know what
It
it is.
is a sample of water is contaminated with mercury 1 oxide. In order to remove the mercury, which is toxic, I have a choice of 3 compounds to add: CaCl2, NaSO4, and MgNO3.
Okay, sorry, read those to me again. CaCl2,
And then NaSO4, and then Mg, NO3, and a bracket 2.
okay, and it was mercury,
1 oxide.
so HgO, right? Mercury 1 or mercury 2?
1.
Mercury 1, so Hg2O. Okay. So, I've got that stuff in the water supply. It's bad. Mercury is not good for you. Okay? So, I need to add one of those 3 things to the water supply in order to get the mercury out. So what I want to do is I want to look at my solubility chart and find a non-metal ion that mercury is not soluble with. Okay? I'm going to eliminate one right now. I'm going to eliminate nitrate. Everything's soluble with nitrate. That's not going to help me at all by adding that. Okay? Mercury nitrate is going to be soluble, so that's no good to me. Okay? So it's either sodium sulfate, okay, or calcium chloride. Now, in general, are sulfates something you want in your water? Sulfates are not generally very good for you, but if it's in a precipitate with mercury, it's going to settle out, right? It's going to precipitate. It's going to be out anyway. So it's not a terrible idea to add that, okay? So what I'm going to do is I'm going to check. If I add sulfate, okay, is it going to be soluble? No, it's going to produce a precipitate. No, it's not. With mercury, that's 2+, mercury 2+. So that's not going to help. Actually, the other one's mercury 2+ as well. So I got a choice, actually. 2 of these will work according to our solubility chart. Okay, I can add sulfate, okay, the sodium sulfate, and I'll get a precipitate of mercury sulfate, okay, and it'll precipitate out, and then I can filter it off. Or I can add calcium chloride, okay, which will also precipitate out the mercury. Okay, now which one do you suppose we typically add to our water supply? Calcium chloride.
That's
Yeah.
like baking soda.
No, sodium bicarbonate is baking soda. Calcium chloride does tend to make your water hard. Calcium makes your water hard. Okay, so places that have to add that, okay, are going to have harder water, but it is something that is typically in water anyway. Okay, and the chlorine we add for a couple of reasons. Chlorine precipitates out lots of heavy metals, so it is typically added, okay, to drinking water. And it also kills lots of bacteria, okay, and other waterborne things that could make you sick. Okay, so calcium chloride is probably the best choice simply because it's one that is going to be in water commonly anyway. The 2 parts of it are not going to hurt you.
Are we supposed to know what type of chemicals different drainage facilities?
No, I'm just saying, this is like for your knowledge later. Okay, what's another thing we add typically to water?
Water?
No. For your teeth.
Fluoride.
Fluoride. Okay, some places do, some places don't. There's this 50-50 split on don't add fluorine to my water. Fluorine's in rat poison, which is dumb, but it's not the same stuff. Okay, and add fluorine to my water because it makes my teeth healthier. Okay, like if you go back to your grandparents' generation, lots of them probably have false teeth. Okay, because they didn't add fluorine to water for most of those years. Okay, and people's dental hygiene was not very good, and lots of people lost their teeth. Okay, your generation, my generation, we have much better rates of retaining our real teeth. okay? Because fluoride has been added to the water and it makes our teeth stronger, okay? So fluorine is another one. If you look at it, precipitates out, okay, lots of heavy metals. It precipitates out strontium, it precipitates out calcium, which is handy, okay? Precipitates out barium, iron, mercury, and lead. Okay, so it's another handy one to add to the water to help get all the really bad stuff
So
out.
we should just be pumping all the water full of fluoride. That's what we're doing.
Most places do. Yeah. Okotoks has gone back and forth in the 20 years I've lived here. It's gone back and forth twice on that, I think.
You should keep it.
Well, there's people that say you shouldn't. Okay, usually though something like that is kind of not understanding what fluorine does in the
But
water.
those people are usually the loudest.
And yep, the people with the least powerful argument are often the loudest. Yeah, it's unfortunate, but it's also true.
The world.
Yeah. Okay. Yes. Exactly. All right. All right. What else? What else are we going over? Okay. So does that one make sense? Okay. Explain the layers of the leaf. Okay. Layers of the leaf. All right. So the top layer of the leaf. Okay. The top layer of the leaf secretes the waxy cuticle. Okay. So. Yeah, here's the yellow line is going to be your waxy cuticle. This blue layer of cells is the epidermis. Okay, the epidermis basically has no chloroplasts in it. Its only job is to secrete the waxy cuticle, which seals the leaf and prevents evaporation. And I say prevents because it does do that. It doesn't control it, it prevents it. Okay, now underneath that layer, that's going to take me forever. I made that upper layer way too long. Wasn't thinking ahead on this one. You see how my cells got bigger as I went along? Okay, so that's the palisade layer. Okay, the palisade layer are the cells that are very rectangular. They're full of chloroplasts. They're packed in really tight. Okay, their primary job is to carry out photosynthesis. Okay, so remember, the layers of the leaf was our example of specialization in a multicellular organism. We could clearly see different types of cells doing different jobs in the layers of the leaf.
What color would a leaf be if it had no chloroplasts?
Dead.
White.
Well, actually, it would be probably what colors it would be in the fall.
Oh.
The chloroplasts stop making chlorophyll, and we see the other pigments that are there. Yeah. Okay, underneath the palisade layer, okay, are the kind of irregularly shaped, okay, cells. It's getting really messy, but okay. Okay, so the irregularly shaped cells of the spongy layer, they also carry out some photosynthesis. It's not their primary job, okay, but they create, because of their irregular shape, gaps, okay, between them that can hold water, carbon dioxide, and oxygen. Okay, underneath that layer is another epidermal layer. Whoops, that didn't change color like I wanted it to. Okay, is another layer of epidermal cells. But embedded in that layer, okay, are those special guard cell things that make up the stomates. Okay, so you'll see those kind of periodically, okay, throughout the lower epidermis. Okay, so the lower epidermis secretes a cuticle. The cuticle covers the entire surface of the leaf except for the stomates. Okay, and the stomates, remember that they can open and close based on osmotic pressure. Okay, so as salt builds up in the leaf, in the spongy layer, because water is evaporating, okay, those guard cells start to have water pulled from them. And when the water is pulled from them, they get straighter, and it closes off the stomate. When there's lots of water available, their vacuoles fill up, and they assume the shape kind of like a bean, which opens the stomate instead. Okay, so another example, those are specialized cells that do a job of allowing water to evaporate, okay, based on salt concentrations. Okay, so layers of the leaf, specialization. Okay, what else?
Could you look at the polar nature of water?
Polar nature of water. Okay.
Is this part of the bio or the?
This was, well, we talked about how it is used by plants. okay, in the biology unit. Okay, we talked about why it happens in the chemistry
One
unit.
right here.
Okay, so. Okay, so we got hydrogen ions here, okay, and then we have an oxygen atom here at the bottom. Okay, so in a water molecule, the electrons are shared because it's a molecular compound, okay, but they are not shared equally because oxygen is more electronegative. Okay, its electronegativity is 3.4. Okay, then hydrogen is. Its electronegativity is 2.2.
Is that what that number is?
That's what that number is. So that number tells you how hard that atom or element holds onto electrons. Okay, so rather than sharing electrons, so if this was the shell in which hydrogen's electron is supposed to go, okay, and it would be just kind of floating around out here, going around and around. Same with this one. Okay, oxygen's outer shell would overlap. Okay, well, what happens because oxygen is more electronegative is these two shells actually end up doing this. Whoop, not what I wanted. They actually end up shifting like this. Those electrons don't even really, they might like slightly orbit the outside of the proton, okay, that is in the nucleus of the hydrogen atom, but they might not. They might just spend most of their time down here. Okay, so what that does is it exposes the positive proton or the nucleus of the hydrogen atom, okay, making that end or those two ends of the water molecule positive. Okay, so there's kind of two positive charges sticking up off the top of the water molecule. Because these electrons have moved so far down, what ends up happening is the bottom two sides of the oxygen molecule end up looking negative. Okay, so now it's polar. There's a positive pole and a negative pole. The charges are differentiated. The electrons are still being shared, but now it looks charged. Okay? And so what water can now do is it can be attracted to other polar molecules. Okay? The positive end of this one to the negative end of another one. So if I had another water molecule right here, okay, these two would create a hydrogen bond between them.
So this is like kind of off topic, but if two like planets that both had like poles came like close enough together, would they create a bond?
No, they would orbit each other. Gravity works different than electromagnetic stuff. Yeah. Okay, so that would be a hydrogen bond. It's strong enough that, you know, it can be used to draw water up the plant or have the water molecules stick to the inside of the xylem, okay, or create surface tension and stuff like that. But it's not that strong. It's easily disrupted, but it's also easily reformed.
Also, a little off topic, why does from dubnium to oganesson not have the,
From Dumnium?
Here?
yeah, why doesn't it
Okay,
have?
so all of these, okay, all of these are man-made. They're produced in a particle collider.
That's so they have like.
They have incredibly short lives. They disintegrate basically. They come apart because the molecules or the atoms are so big that they can't stay together. So they don't get a lot of time to study their nature before they're gone. Yeah.
Also, why does borium have a lower mass than C-borium, but it's like a,
What are you looking at?
Oh, yeah.
like the lower mass of borium and C.
106 and 107.
106 and 107. Honestly, couldn't tell you. I mean, you got a 268 and a 277 here. Okay. Couldn't tell you. These, once you get into here, all these man-made ones, they're very different. They behave very differently. Again, they don't get a lot of time to study these. It's a flash in the pan and it's gone. Yeah, sure. Okay, so does that explain the polar nature stuff? Okay. All right, what else?
Different.
The bigger vector form.
A vector.
Yeah, the vector.
Okay, so I'm not gonna have a V equals, like a big V equals D over T kind of question, but I might put something maybe in the multiple choice that might be something like this, okay? What Persum? Persum, that's the thing.
Is there any question we will have to tell how many like protons?
Okay, I'm in the middle of this one.
I like math.
Someone asked about this one, I'm in the middle of this one.
Yes. Okay.
Okay, so you could get something like this, okay? This would be a pretty simple calculation in the multiple choice question, okay? So a person walks 50 meters north, then turns and walks 30 meters south, so I'm giving you directions, I'm giving you vectors here, okay? The whole trip takes 60 seconds, so I've given you the time. What's the average velocity of the person? Okay, is velocity a vector quantity?
Yeah.
Okay, so now those directions mean something, right? Okay, so if I go 50 meters north and then I go 30 meters south, what's my displacement? Because if I want to find velocity, that's displacement divided by time, yes?
20 meters north.
20 meters north. Okay, that takes 60 seconds. Okay, so 20 over 60, okay, is 1 over 3, so 0.33.
That's not very fast.
No, it's not very fast, but it's a vector quantity. Okay, I could have just as easily given you all that information and asked, what's the speed? What would you do differently?
Not right down north.
And
Just add, right? My total distance would be 80 meters, 80 over 60, okay, and I get a very different number. Now, if this was a multiple choice question, okay, this is probably what I would put in there, okay? First, I'd figure out what 80 over 60 is, okay, and I'd go 1.23, so I'd go A, 1.33 meters per second, B, 1.33 meters per second north. Okay, and then I would probably put in C, 0.33 meters per second north. Okay, and then D, 0.33 meters per second. Okay, remember how I said I'm going to figure out how you could do it wrong? Do it that way and put that answer in there. Okay? Because if you think this is scalar or you do this like it's scalar, you're going to think you did it right when you didn't.
My brain does not like all of
Okay.
this. Yes. I'm definitely gonna pick like.
I would pick better
these,
numbers, but
try
yeah.
and be
Okay, so something like that. Definitely. That's where, and I would ask it in the multiple choice. I'm not going to burn a 5 mark written on something like that. Okay, is that good? Okay.
Could you please do something on the hardest mole equation?
Okay.
Yeah, no. The tricky ones. Dude, you just tricky.
Okay, there you go. Give you a few minutes on that one. Okay, so on this one here, we're looking for little m. We're looking for the mass. Okay, we're told the number of molecules, okay, and that is 8.26 times 10 to the 26, okay, for the number of molecules, and we're told the material, okay, it is calcium nitrate, okay, but when I drop and swap, calcium's a 2 plus, nitrate's a 1 minus, so it's NO3 2, okay, and guys, that's the mistake people make on a test. They do not check drops and swaps. I'm gonna make this ionic compounds, okay, you gotta make sure you do your drops and swaps correctly. All right, so we're gonna have our 40.08 for our calcium, okay, plus 14.01 times 2 for our nitrogens, plus 16 times 6 for our oxygens. All right, so we're looking at 164.1 grams per mole. Okay, for big M. All right, if I'm going to find M, I still need the number of moles, and I wasn't given that. But I was given the number of molecules and Avogadro's number, which is the number of molecules in 1 mole. Okay, so what do we need to do with those two numbers to get the number of moles? Divide them. Okay, if I go 8.26 times 10 to the 26 molecules, okay, and I divide that by 6.02 times 10 to the 23 molecules per mole, the molecules are going to cancel and I'm going to be left with moles. Okay, so 8.26, 26 E26, okay, divided by 6.02.
You didn't put the divide.
Oh, I didn't get the divide in there. Oh, come on. Divided by 6.02 E23. All right, so is that a large number of moles?
Yes.
Yeah, it's going to be an even bigger mess. Okay, so now that I have the number of moles, so now I have N, 1,372 or whatever it was there, okay, moles, can I now calculate the mass? Okay, so N equals little m over big M. Okay, if I'm looking for little m, okay, I multiply both sides by big M. Okay, so that's going to be 164.1 times 1,372, and I would include all the decimals when I do that calculation. Okay, so times, and then 164.1. Okay, so that's a lot. Okay, it's 225 kilograms. Okay, yeah, roughly, like, well, actually it's a little more than that, but yeah. Okay, so like half a ton. Yeah. Okay. So 225. Okay. 225,160.47. It's a lot. Okay. It's a big number, but that's the number of grams. Quarter ton. Yeah.
It's like my
It's
warm
2,000
lateral.
pounds. Yeah, ton. Yeah. Okay. Making sense? All right. What else do we want to do? Active transport. Yeah. Okay, so in active transport, if this is a cell membrane here that I'm drawing, okay, in a cell membrane there are transport proteins and they can change their shape. I don't even know how to draw those. Do something like that, and then a similar shape on the other side. Maybe I'll just do this.
Just draw telephones.
Yeah, draw a big old fashioned.
Clone it, and then I'll tip it. Yeah, all right. Okay, so they look kind of like this. It's terrible, okay?
It looks like two beans facing each other.
That's what it's supposed to look like. Okay, awesome. Okay, so what happens in active transport is, okay, inside the cell, I already have a high concentration of something. Outside the cell, I have a low concentration of something. Naturally, things want to go from left to right, high to low. Okay? In active transport, the cell uses energy to change the shape of these two molecules. So they go from being like closed like that, okay? And then when the cell applies energy to it, they change their shape so that they're like open and accepting stuff in, and then they pump it through to the other side. So they just keep changing their shape like this. It's terrible animation, but okay, they keep changing their shape like that using energy from the cell, and they're pumping that material against the gradient. The material would want to go the other way, but the cell is using energy to move it essentially uphill or up the gradient. Okay, so they use energy, they move material across the membrane against the gradient, and the carrier proteins change their shape to make that happen. Those would be the 4 points I would be looking for in a question about active transport. Okay, same as what was on your unit exam. Okay, when I asked one about active transport, I was looking for uses energy, goes against the gradient, okay, across the membrane, carrier proteins.
That one was awesome. I just fully described what the picture showed me.
Which was exactly what you were supposed to do, but the picture is way better than that one. Okay, you may. Okay.
Can we go over like endo and exocytosis?
Okay, so here's all you need to know about endo and exocytosis. Endo is into the cell,
Yeah,
exo is out of the cell.
but then there's the processes that go with it too.
Right, now those are incredibly minor,
Minor.
okay? All you need to remember is which one is which, okay? There's not gonna be any understanding of the mechanics of them,
Okay.
okay? So you've got phagocytosis, which is eating, okay? Cell eating, so the membrane wraps around a food particle and draws it in.
Okay.
Okay, now mechanically what the membrane does is the same for all three processes. The membrane engulfs something,
Yeah.
okay? It's the reason or the material, okay? So phagocytosis, it's food. Pinocytosis, it's water, okay? The cell is like very dehydrated and it's willing to expend energy to change the shape of the membrane and engulf a whole bunch of intracellular fluid, okay? So that's pinocytosis, okay? And the last one is probably the most common one, and that is receptor mediated. Remember how we talked about how the membrane is covered in all those little receptors, the little antennas? Okay? If one of those antennas or receptors gets triggered, okay, by the hormone usually that bonds to it, it will tell the membrane engulf it right now. Okay? So that's receptor mediated endocytosis because it's the receptor that causes it to happen. Okay? So mechanically, it's all engulfing. Okay? All that's different is what's getting engulfed for what reason.
Alright.
Okay, so cell eating, cell drinking, taking in a hormone.
Could you please include like plant transport?
Okay, plant transport. Okay, here's what we essentially need to know for plant transport. Okay, so you got your stem. Okay, near the outsides of the stem is the phloem. That's where the thick, sticky, sugary sap is, okay, in the phloem. Okay, in the phloem, okay, you have sugar, and it is moving downwards. Okay, what's making the sugar move downwards?
Gravity.
Okay, that's what usually makes things move downwards. Okay, so the phloem is moving sugar, okay, and it's using the force of gravity to do it. Okay, in the middle, okay, are millions of microscopic tubes, okay, called the xylem. Okay, so right now I'm making it look like it's one big tube, but it's actually millions of tiny little tubes that would take me forever to draw. Okay, so the xylem is in the middle. Wow, that's not even close to the word xylem. Got ahead of
I think
myself.
I've spelt it like that multiple times.
Xylem. Okay, okay, so it's in the middle, okay, and water is moving up. Okay, so the xylem has got water. Okay, water is moving upwards. Okay, the forces that are moving water. Okay, so if this is a water molecule here, and this is another water molecule here. Okay, and this is another one here. Okay, there are hydrogen bonds between the water molecules. Okay, that's cohesion. Okay, there are hydrogen bonds between the water molecule and the wall. Okay, that's adhesion. Okay, and then there's this force here.
I just wanted to say another easy
that is the osmotic pressure from the spongy layer in the leaf. Okay, so if it's a 6-mark question, what I'm looking for is water, xylem, osmotic pressure, polar nature of water, phloem, sugar, gravity down.
If I just write all this little key.
Quite frankly, if that's what you wrote, that's what's on the sheet, right? Okay, I mean, be a little more eloquent than that, but okay, that's what you need to know. Those are the basics of transporting plants. The water is transported in the xylem, it's pulled upwards by osmotic pressure, which is caused by water evaporating and leaving salts behind. That's kind of an important part of the explanation, okay? And then you've also got the polar nature of water, keeping it from falling back down, okay? And then in the phloem, you've got the sugar that's being pulled down by gravity. It's thick and sticky, okay? And it's on the outside.
How does transpiration occur?
Transpiration is just evaporation through the stomates.
And how does the guard cells do that?
So the guard cells work by osmosis. So in the leaf, as water evaporates, what gets left behind? Salt. Okay, so that salt is what's creating the upward force for the water, but its buildup is also going to start drawing water out of the guard cells. Okay, when it gets to a certain point where there's no more water coming up or it's not coming up fast enough, it draws water out of the guard cells and closes them, and then the plant stops transporting water because it's obviously getting stress. Yeah? Can we do that later?
Could you do like an example, like a graphing question?
Oh, that's gold. That is gold.
Hang on, I'm gonna find. Okay, here's a couple of graphing questions.
Thank you.
Okay, you got a position versus time graph. It has an equation.
Yippee!
Okay? And this question might ask, did we do this one yesterday? No. Okay. I feel like it was like really recent that I did this one. Okay, so on this one here, first thing I might ask is, what's the velocity of the object?
2.7 meters per second.
Positive 2.7 meters per second, because it's vector, right? Okay, the slope of the line on this graph is the velocity, because it tells me the rate at which y, position, changes as a function of x, time, meters per second. Okay? All right, second thing I might ask. What position will the object reach in 25 seconds?
That was a dumb one.
No one said you had to manipulate it to solve that one.
25 seconds.
If we don't have to.
Yeah, 3, 2 points.
Yes, yes.
67.
25.
67.
Okay.
68.
Bit over a minute.
You guys are just always my stuff, so it doesn't.
67.8.
Sounds about right.
Bit over a minute.
Yeah.
Would I get in trouble if I just said a bit over a minute?
Yes. Well, you won't get in trouble, you just won't get the marks. Okay, so if I'm looking for position, what am I solving for, y or x? Y, because position is on the y-axis. Okay, so all I have to do then is go y equals 2.7 times 25, they gave me x, plus 0.309. Okay, so punch that in, 2.7, okay, times 25, plus 0.309. Okay, 67.809, so. And that would be positive. Okay, another thing I might ask. How long to a position of 100 meters?
That one you have to manipulate.
Yeah.
6.9.
Okay, it's asking me how long. It's asking me to solve for time, which is x. Okay, so I've got to take y equals m times x plus b, and I've got to manipulate for x, so I subtract b, then divide by m, and I get x, so that'll be 100 minus 0.309, okay, divided by 2.7. Okay, you know what, you don't have to be here.
It's spilled.
No, it's spilled because you're horsing around.
No, it's spilled because I put my elbow back and it's right behind my elbow.
You want to leave, there's the door. Okay, so it would take 37 seconds. Okay. All right, so that would be position versus time graph with a linear function. It'd be similar if I gave you a velocity versus time graph with a linear function. I just would ask you how fast at this time and how long to reach this velocity. Okay, and instead of asking you what's the velocity, I'd ask you what's the acceleration, and it would be the slope. Okay, so similar function, just okay. If it's on a velocity time graph, you're solving for different things in the same way.
Can you quickly go over the tropisms?
I got to do the velocity time graph. They asked about both kinds of graphs first. Okay. Velocity time graph. Okay, so here's a couple of things I might ask. What is the acceleration between 0 and 5 seconds? So this is 5 seconds right here.
Would that be?
It's 8, I think. We'll say it's 8.
Is that line not straight or is that just?
It's not straight. Well, it's two straight lines. It's not one straight line.
Oh, it looks close enough.
And this is -4. This is -4 here.
What's the acceleration? What's the acceleration?
Okay, do we have a formula that solves for acceleration? Okay, what is it?
A equals BF.
Can I use that here? Whoop.
Yeah.
Okay, so VF is 8. Okay, VI is -4. And the time is from 0 to 5 seconds. Okay, so 12 over 5, 2.4. But it would have to be positive 2.4 meters per second squared. Okay, one of the things I might ask. Second thing I might ask, what's the displacement of the object between 2 seconds and 9 seconds?
Wait, is this where you do the little point? 4 times 8 is 4.
Okay, ready to go over it? Just for the sake of time?
Yep.
Yeah.
Okay, I gotta find the area of those 3 shapes, yes?
Yep.
Okay, so this triangle here is 3 on the base. It goes from 2 to 5 seconds. It's 8 high. Okay, 3 times 8 is 24. It's a triangle divided by 2. Positive 12 meters. Okay, this rectangle here is 2 on the base. It's 8 high. It's 16 meters. Positive. Okay, and then this triangle here is 8 high, 2 seconds wide. It is positive 8 meters. Okay, so that's 24, and another 12 is 36. Positive 36 meters. Now, what if I had included that other triangle that's on there? It'd be negative, right? Okay, so I'd have to make it negative when I added them all together. Okay, last thing, tropisms.
Yeah.
3 of them. Phototropism, geotropism, gravity, thigmotropism,
That's the vines going up.
touch, right, could be wind too, right? Okay, so important to know, guys, those are the 3 tropisms, okay, and we got like a picture on the test that we had to explain. Expect something similar.
And then the auxins are what then?
The auxins make it grow faster on one side, yeah, you gotta explain the mechanism.
Are auxins the only?
Yes, for the tropism, yeah. Okay, so make sure you know the mechanism. Okay?
Like that's the only hormone that I
No, you have to know all the hormones, that's the one that makes the tropisms happen.
I have to write another.
Okay, guys, thanks for a good semester.
What room is it?
St. John, I think Miss Olszewski's are, okay? Make sure you bring what you need, okay, to the test. I will pop in periodically, okay, to check on you, because I'm not supervising you, okay? But I will pop in periodically to check on you and see how things are going. If you have questions, you can ask at that time, okay? All right, you're welcome.